First question, Jim.
Yeah, I'm in awe of all this. It's great. And I'm just a country doc from Alabama, so if this is a naive question, please bear with me. I'm curious, I see you have analyzing 4 databases at a time. Why you don't combine the populations to get a narrower confidence interval? Because there were some of those graphs you display where I was looking going, boy, you know, if you brought them all together and got a narrower confidence interval, you might find something significant where it's really not a question of is the effect size too small, is the population too small?
It's a great question. I'll take that.
Okay.
So there's a couple components I want to talk about. One is the logistical of the fact that as a distributed data network, everybody's got their own data, and what they can do with their own data is only produce an estimate from each source. And so as a distributed network, one of the ways we want to function is to be able to share results with each other without actually being concerned about patient privacy or sharing data. But actually, I'm actually more interested in the methodologic issue, which is when you combine data, you're making a very strong assumption that the data are in fact comparable. That is, that they can be brought together. And what I was trying to show in my characterization is, in fact, what we see is every time we look at 2 different populations, we actually see appreciable differences in those populations. And so pooling data actually is obscuring what is in fact potentially very meaningful differences. And so what I would actually advocate is that we need to expose those differences. When we produce a forest plot like that where we show every data source specific thing, your eye was led exactly to the right point. Maybe despite all of the database differences, we still reach a consistent answer, and to George's point, like we're on a constant surface, and that will be great. But we want to be able to make sure that we account for those differences when we try to answer the question, rather than assume there's no difference and then be tripped up at the end.
Thanks.
Very impressive talk, all 4 of you. I have a question for Martijn. At the end, or the whole vision was to, at the end, to distribute it to patients. And I was really impressed when you keep extending the scope until you extended it to comparing all the drugs for depression against each other. I think only pairs, so not really more complex than just one against another. And I was even impressed there was a procedural intervention. So can we, after you adjusted even the intervals, you ended up with 11%, which were significant. And if I got that right, these would be the ones which we want to let the patients know. Maybe I would be, let's say I'm trying to define whether electroconvulsion therapy is correct, and maybe one of those golden nuggets are telling me a story about that. So is that correct that these would end up being distributed to patients? And are you really confident that we can just say, we're going to get claims like, based on your advice, I did that. What is that last piece for the patients?
Yeah, so thank you for that question. And I didn't touch on that because I don't really have a good answer for that yet. So now that we've generated all this data, how do we get it to the patient? One thing that I think you touched on is the idea that what if we focus on the things that are statistically significant? What you're then doing is you're doing an exercise known as signal detection, which is something you could do with this data set that we've generated, but now you should really start to think about the multiple testing that's implied there. If you really look at all 24,000 estimates and only set a cutoff somewhere, that can be problematic in that you generate spurious results. What I think would be probably more helpful for a patient is for the particular question that they're interested in, if it's like 2 different drugs or 2 different treatments that they're considering, only for that comparison, present them with the information that we have for that, including the ones that are not statistically significant or very suggestive of actually there being just no effect at all, and also presenting that and adjusting only for multiple testing as we present it to that particular patient. Hope that answers the question.
I mean, there's a trick. If I aggregate across 100 patients I see, I eventually will end up building that knowledge base of advices I'm giving. So it mitigates the multiple hypotheses, but over a large population, I will end up giving individual advices. But that's a good approach, and I agree it's a hard topic. So.
I do want to just add, though, I think we all need to stop with the notion that the only things that matter are statistically significant findings. I will argue that every one of those every study matters. So every dot on that plot, whether it's significant or not, matters. They're equal value, they're equal evidence. It's just as important to know that sertraline does not cause stroke relative to duloxetine as it is to know if it causes ingrowing nail. We should not protect the patient away from information that gives them insights of their care.
Hi, Robert Carroll from Vanderbilt. I had a question around the negative and positive controls. It seems like the negative controls that are being selected are not necessarily hypothesis-free, and then you're generating from that the positive controls. I wonder if there's any reason why you don't artificially simulate negative controls, for instance, or find some other way. There may be real signal that you're not able to find in your particular studies or your particular populations. I was just curious why you chose that pipeline, or if there are other ways to assess the same to still be able to normalize your odds ratios and relative risks.
So the nice thing about using real negative controls is that they may have confounding that is representative of what you'd have for your hypothesis of interest that is outside of what is captured in your data. So using real negative controls, you will be able to detect whether or not there are unmeasured confounders, whereas if you use fully simulated negative controls, just because you're simulating it in the data that you have and nothing more, that means that you're actually liable to basically overestimate or underestimate the bias that's inherent in the study. But you're absolutely right that our negative controls do have the problem that the assumption has to be right, that they're negative controls. One way that we try and be at least a little bit sure about that is that we only include exposures and outcomes where at least for the exposure there's at least one evidence that the exposure causes something, and for the outcome there's at least some evidence that the outcome is caused by something, although of course it doesn't have to be that the outcome causes exactly that outcome. So we know that we have evidence, we should see evidence if it's out there, but we don't see it for the negative controls. That makes us a little bit more confident that they're really negative, although there is always the problem that they could still be positive, but we don't know about it. That's certainly a limitation. Yes.
Sure. Okay, thank you.
Check out Anthony Senna's demo on how to create negative controls in 15 minutes.
Judy Rakusin from FDA. First of all, I come to this meeting every year because I just want to be bowled over with the great strides you guys are making. It's really impressive. But it all hangs on sort of one thing that I think, Patrick, you alluded to, but that I'm not sure we covered in depth, and that's that we're assuming that the coded data is that the patient actually has that condition. And I think many people in the room are aware that that's often not the case, and that there are clearly settings in which we don't have confidence that the codes are, actually represent the conditions. So I suspect you're working on some automated approach to dig into the validation question, and I'd be interested to hear what your approach to that would be. Because I think as impressive as all of this is, it really hinges on, are those cases of acute liver injury really that, or acute liver failure, or suicidality, or libido decreased, or intentional overdose, or whatever your question is.
I mean, if only there was someone standing 2 feet behind you who is an expert in developing an automated process for phenotyping. So actually, you should talk to NIGM about some of the work that we are doing as a community. What I will say, though, so I agree with your point completely, Judy. We need to be cognizant about the fact that our cohort definitions have the potential for measurement error, and we actually know that the measurement error in some of these cases is big. One thing I'll point out about what both Peter and Martijn showed is that if you apply an evaluation framework, you are measuring error. Measurement error is a type of error. So when Martijn says we are confident to 95%, and you saw the confidence intervals get wider, one of the reasons the confidence intervals got wider was because there was measurement error in those outcome definitions. Another reason is confounding, and another issue is protopathic bias, and all of the thousands of things that we can all regurgitate. But one of those sources of error is the measurement error. And so by using an evaluation framework, whether you have a perfect definition or a completely crappy definition, the evaluation is going to tell you how reliable your evidence is. And we should actually take great stock in the fact that you might go in with a not so certain about a definition, but if the evaluation tells you you can trust the evidence, that should give you some assurance that even though you weren't so positive about the definitions, you still might be able to learn something meaningful.
Exactly. The negative controls will find average coding bias. You would still worry that the condition you're worried about has differential coding bias, and that's why we'd have to look at it intelligently. But the average coding bias will be accounted for.
Thank you.
I'm Ray Park from the Aju University in Korea, and I have one question for the Peter and one question for Martijn. For Peter, you used the three algorithms to make the prediction algorithm, and I think it's a very light way of the algorithm instead of the hardcore algorithm, such as the artificial neural network or the SVM or deep learning algorithm. And it is known that, for example, the deep learning algorithm have a 10% higher performance than the other algorithms. So is there any plan to use that kind of the heavy algorithm in your future plan?
I knew somebody was going to bring up deep learning, but I didn't think it was going to be Ray.
No, I'm surprised. Well, the answer is actually yes. We try to look at all those algorithms, and actually, while I was in New York at George's group, I had the opportunity to visit NYU and met with David Sontag, and he's an expert in doing that deep learning work, and we really want to go into that. And I'm actually looking for a postdoc who's going to do that work in Erasmus, especially that type of work.
Okay, another question for the Martijn. You made the automatic system doing all kinds of research for all kinds of database in a very short time. If you suppose that kind of the system make research every day for 100,000 research every day, then what will be remained for the researcher's future? That what I mean is, do we have maintain our job as a researcher in the future?
Well, so as I was trying to make clear with the analogy with the Large Hadron Collider, I don't think we're out of a job. I think what our role should be is not producing the one estimate at a time. I think we should all think really hard about that process that we're using to generate the evidence. So I think that's where our efforts should move.
Thank you. To mention, we have some deep learning experts in the audience with us today, sitting Rajesh, David, so on. So we, if you have any, want to ask any questions about it during the next session, I can point you out to them. Next, David.
So one of the things we say in our auditioning mission is to generate evidence that matters to patients. And I think we should have a mechanism for getting questions from patients. What are the conditions for which they want evidence, instead of us trying to second guess that. So that's sort of a comment. And it relates to getting the patient-reported outcomes and all that stuff. But the most important thing, let's get questions from them. And then the second thing is, drugs are great, but they're a small slice of the $3 trillion in the healthcare system. The questions that make big impact typically evolve around surgical procedures. What's our path as a community to reach there and get those groups, like the device groups, engaged and involved? So 2 questions. How do we get the patients, and how do we get people outside of drugs?
I'll take the second one because it's easier. So one thing we've been very deliberate about is to generalize the analysis problems as we just described. It should have been clear to everybody, we did not develop tools to study drugs. We developed tools to study interventions. We did not create tools to study diagnosis codes. We created tools to create cohorts and phenotypes. And the analysis, whether you're doing a prediction is just a population at risk defined by some outcome in the future. And if you're answering a comparative effectiveness question, it is simply one intervention compared to another for some outcome. Those are very deliberately and strategically developed so that we can support whether your question is drug versus drug, or whether you should go check out Ajit Landi's poster. He's talking about a drug versus a surgical procedure. If you are in a device business, like some people in the room, if you just want to study interventions of devices, the OHDSI stack is fully supportive, and we're the only data model that explicitly contains device exposure information to enable that. I'll leave the harder question for everybody else.
Well, I can take the first one, actually. So regarding reaching out to those user groups, I think that's a very important thing that we should actually do. And in the European Medical Information Framework project that we do that we have in Europe, we have people from the Alzheimer's and diabetic research area. And I've reached out to them and asked them the question, what would be prediction problems that you would like us to focus on? And I think that might be one of the opportunities that we can leverage, and ask them for a whole set of prediction problems, then we focus on those. Besides going from the data approach, I think we should do both efficiently.
Certainly it's in our dream to be able to answer those questions. Remember, we do want to do the public RFA. We ask everyone, what do you want to know? And we're still in the process of building up the infrastructure so that we can push that button and have the whole thing go. But we definitely, that is exactly our, you being part of it, our goal.
So the main reason I'm asking these two questions is that every time I ask these questions at this hotel, something happens in 2 weeks. So, it's a little bit superstitious, but I think I should still ask the question.
Thank you so much. I have been following your work very closely, and you know how much I admire the progress that you are able to make. I learned as a bench scientist that you need to use all the controls that experiments require. I also know that in clinical pathology, you use as many controls as possible. Then I think that we need to have a different type of controls. These are the positive controls that are biologically plausible and that they appear consistently across other members of the class. And we have multiple examples, like, for example, cough and angioedema with ACE inhibitors and ARBs, gum disease with calcium channel blockers, for example. Then we need to have we need to include, and I understand that in many instances, maybe they are not collected in the data, but we need to understand this non-collection. I think that we need to have always a set of positive and negative controls that are biologically plausible. And this is where safety data mining, the MGPS method that is not data dredging, has shown that can help actually a lot. And it's not our findings at FDA, but the GSK findings of an association with molecular function, etc. Just a thought.
Yeah,
Thank you.
So I do want to clarify one thing, and thank you, Anna, for making that comment. So I think as a community, we all need to be very careful in the language that we use to describe what we're doing and why we're doing it. So I want to make it very clear that what I presented was not data mining, not because I think that there's anything negative about data mining, quite the contrary, we're doing lots of data mining, but what I was doing was just simple observations from a data set. What Martijn was doing was causal inference, using the latest set of causal inference methods. That's not data mining. What Peter showed explicitly was machine learning. That is the latest in data mining, the latest and greatest in large-scale data mining. That is data mining. So we just need to be clear that just because you see a lot of numbers does not mean you did data mining. And just because you do data mining doesn't mean that it's somehow a lower quality of evidence. but rather, when we generate evidence, we need to provide a clear understanding of what the scope was, what was the intent, and then how can we interpret the results to understand their relative validity. And whether that's characterization or prediction or estimation, I think that that same framework can hold for us to know how much we can take stock in the information we've produced.
No, I agree.
I mean, I'd also mention that I don't think we're done figuring out how to do positive controls and calibration. And I think, so Martijn, Mark Suchard, David Madigan, Patrick, and others from that team that I didn't mention just now are working on it. So I think, actually figuring out how to use a positive control where we don't know the exact positive number, but how do we use that to adjust our calibration is something we just haven't invented yet, but it's something that may be coming. So I think that it will be important to use positive control. Also, how to use positive controls to test whether we're finding the ones we should be finding is a more straightforward test of our system.
Yes, but fine.
Shout out to check out Erica Voss's paper that's going to be published shortly, which talks about how to build a predictive model to find biologically plausible positive controls.
Very good.
Okay, thank you.
So thank you. I think the comments that you made, Martijn, regarding the publication issues, p-values, and our adherence to less than 0.05, Ken Rothman and Charlie Poole and a lot of others have talked about this for a very long period of time, and it's nice to see you trying to make that empiric. I guess I wonder, though, you showed all of these drug outcome pairs where there was no effect, and I think we have to be cautious about saying that there's no risk with medicines, right? We're assuming you put up this idea of a causal construct. The causality framework, have we really done that? Because I think when we say drugs have no risk, just like when we say they have risk, that has real importance. And the null finding, although it doesn't get out in the literature as much, you showed a lot of things that have no risk. And I think we need to be very careful as a community about saying things have no risk. And when I looked at some of those outcome pairs, did you really do all of the sort of causal diagram theory and think through all of the issues with each of those outcome pairs? It seems to me like there might be a lot of bias to the null in a lot of what we do because we measured use, not use, or use of one versus the other. So I guess when I look at this, it's amazing what you guys have done mathematically, but also there's a motive piece and there's a trust piece that we want to bring forward. And when you look at that picture, do you feel confident to say to the patient that there's no risk? Because I think that's ultimately what we have to do. And I think as a community, academia, biopharma, the government, that's really what we want to get to, is the right answer for the patient. So I don't know if there's an answer to it. I know it's not an easy question, but do we feel confident to say that there's no risk for those 50,000 pairs?
Well, so maybe I wasn't very precise in my language, but I think we're all in agreement that failure to reject the null is not evidence towards the null, meaning that one of the reasons I like the fact that we now have a mechanism, even though it might be imperfect, for calibrating the confidence intervals, is that just a failure to reject the null should never be communicated as saying there is no effect, because it just says we don't have evidence to support that there is an effect. There might be an effect, but the confidence interval tells you much more about the uncertainty we have where the effect might be. And so certainly I would not be suggesting that we focus on things that are statistically significant or the p-value at all. Actually, I would like us to focus on these confidence intervals, and some of them are remarkably wide because we have bias as measured through our evaluation.
Yeah, so I that's very well spoken. I guess the thing is we talk about getting this to patients. I wonder how many patients understand p-value calibration. Right, you may, Patrick may, others may, but we're going to show them a plot of everything where there's no risk. I think that's a very tall order. And so I just throw that out for your consideration as we think about the promulgation of this information.
It's also important to communicate to the patient. We are not saying there's no risk. We're saying to the patient that we can't tell that one drug has a higher risk than the other drug. So they're still at risk either way, potentially. But when they're choosing two treatments, at this point in time, we can't tell if one drug or another is going to cause a greater risk of that sort. And I think to the patient to know that well, it could be still susceptible to stroke, but whether I pick this drug or that drug won't change that susceptibility. It's a different thing to communicate than saying to the patient, don't worry, you're at no risk for stroke, which in fact they're always at a risk for stroke even without the drug. Number one, they have to understand that. And number two, if they add on one of these drugs, either one, there could be an increased risk of stroke. We just can't tell if one's greater than the other, in part of them making the decision between those two.
Yeah.
Hi, I'm Christoph Lambert. I'm from the University of New Mexico School of Medicine. Really appreciated all the presentations, and of course I've been a big fan of this approach we're taking of looking across many different questions, dealing with multiple testing, different parameters, different methods, and getting a distribution of the evidence. It seems like the correct way that one ought to be doing large-scale observational studies, the way of putting the evidence in context. And what's been highlighted about the challenge of these one-at-a-time studies is, as was done by Martijn, is how we're doing a lot of overfitting, doing a lot of p-value hacking, if you will. So the question is, what are the implications for all those who do these one-at-a-time studies? Is this going to become the main way that that studies all should be done? Is there still a place for those individual studies? And what's going to happen to those who don't have the capability of doing these things, but their research is sort of the one at a time? And I'm talking particularly about observational as opposed to, say, a small randomized trial.
Well, so I very deliberately tried to frame publication bias and p-hacking as a dissemination problem. So if somebody has a specific question, and for that specific question they need an answer, and they do the study, and they consume the evidence that that study produces, there is no dissemination problem. So if the FDA has a question and they order somebody to investigate that, and they look at the result, at that point there is no publication bias or p-hacking. So I certainly think that there is still a place for the one-off study. I'm just saying that as a field, we should probably be moving towards generating it more at scale if we want to avoid this dissemination problem that is in the literature.
And I'll just comment to the second point. So if you are sitting out there and you currently do one study at a time off of your individual data set, and you're saying, oh crap, how am I going to do this? Join the journey. The whole reason we have this community is because we don't expect everyone in this room to have all of the statistical expertise, all of the epidemiologic expertise, all of the informatics expertise, and all of the clinical expertise that is needed to do large-scale research. Just like nobody could build the Large Hadron Collider on their own. If you are sitting there thinking, hey, I've got some data, I've got some questions, and I've got some thoughts about how to do research, this community is here to work together towards reaching that common vision. And so from my perspective, I think don't go off and try to do this on your own. We couldn't have done it on our own. We had to work as a community to get to where we are. And so for those of you who are on the outside looking in, this is an opportunity for you to join the fold and become part of a collaborator so that you can contribute to this overall process.
I mean, you could argue, taking a softer stance, that if you get a lot of individual studies that are off the surface, I'd be worried whether the surface was right, in addition. We have time for these last two questions here.
Hi, I'm Melanie Blank. I'm a medical officer at FDA, and I'm used to analyzing large-scale clinical trials, drug trials. And I'm new to this, so it's very exciting, but I can see some areas that are concerning to me in terms of the conclusions that you could be making based on comparisons of drugs that have different indications. So therefore, there could be different populations for these drugs. So, for instance, with the sertraline and duloxetine, you'd expect that duloxetine, which is for peripheral neuropathy, and I don't think sertraline has that, you'd expect an older population who are going to have different kinds of adverse effects. So I don't really know that it has anything to do with the medication at all. It's more who's getting prescribed this drug. The other thing is, what if there's like dry mouth or something like that with one of your drugs, and the compliance is terrible, or one is BID and one is QD. The compliance will be bad for one versus the other. So how can you compare those two? So I'm concerned about the ability to really compare this way.
So trying to answer that question, so you mentioned different indications, but to be clear, the analysis that I performed are all nested in a group of people that have a diagnosis of major depressive disorder and not a bipolar disorder or schizophrenia. So we try to handle that specific problem, first of all, by nesting in people that have the outcome that we think is the indication that we're interested in. Also, even if the populations are different, we would expect that, well, we would hope that our analysis approach can adjust for that. If it can't, then that's exactly what the evaluation is for, using the negative and positive control. So we did indeed see that these populations are different, very different, based on the propensity score plot. And we also saw that in this case, for the particular example that I showed, we were able to adjust for those differences, but only within the group that we nested in, the people that have the primary indication that we're looking for. So I hope that alleviates some of your concerns.
There could be comorbid conditions that basically make a clinician decide upon one drug versus the other.
Right, but that should be in the propensity score model, right?
If you say so.
Well, what happens is he does the propensity score matching to try to match them up. If they don't match up, then the diagnostic shows the pink on one side, the blue on the other side don't come together in effect. And what that means is it's possible for us to tell that they aren't similar. Now, what you could also raise is, well, what if it's hidden from us? There are covariates we're not thinking of that differentiates the two that we're not measuring. By measuring 50,000, we're trying to get as many as possible, but that's why it's still research. So, I mean, we still need to do more work. This is our first all-by-all experiment.
And what about the compliance possibility?
Same thing. So compliance is something that you can characterize, and then as part of your estimation, you can try to adjust for. And the question of, are you doing it adequately, that's the evaluation question, is to prove that you have adequately adjusted for all of the factors that make those groups different, just as you would hope for in a randomized clinical trial.
I mean, there may be compliance issues that you're not sufficiently measuring, and that's what we need to look at first. So that's what I would look at. Yeah.
But so not to advocate for controls as the end-all solution, but most of these problems should actually become apparent when you include the controls in your analysis. So,
last question.
Thanks. A comment and then a kind of a question for you guys on your thoughts. So the comment is, it's really amazing that you put this large-scale way of doing these analyses at all by all, as you've described it. And the comment of the last person really makes sense when you talk about propensity score adjustment as being really affected by the information that's available to you in the data set. But when you show a picture like what you've done and you describe it, some other ideas come to mind. I wanted to ask what your thoughts were about this, just one idea. That when I work on, for example, fall risks, and we have a small data set, and we know that some things aren't measured in that data set or measured really well. And so we'll use things that are well known in the literature and say, well, these things definitely need to be in the model in terms of adjustment. When I looked at your big picture, I thought, well, there's going to be some data sets where you ran your method on, and your propensity score did a great job of adjusting for bias. Could you take that information and input it into a data set where maybe you're not good at good distinguishing ability and run the cycle there? Would that be possibly a way to adjust something?
So I think what you're talking about is transportability of
it's like rule transfer in a sense, maybe.
Yeah.
It's sort of an analogy.
That's something I don't think has ever been done before, but it's basically combining what I said together with what Peter said, because the propensity score is basically a predictive model.
To me, it's like when we do the one-off studies, the clinical expertise and what's known in the literature comes into mind when you build your model because of the information problems that you have, right? But you're showing that for some data sets, you're getting good adjustments. So maybe that becomes sort of by analogy something that you could input and run again in a data set where it's not so poor.
My second question, and this will get us to lunch, reproducibility. Reproducibility is really strongly emphasized for us NIH-funded investigators right now. You're talking, in a sense, about reproducibility because you're advocating for making all the methods public and all the information about our data sets and doing the negative, getting our negative findings out there. But there's one thing that just we're continually not able to do, and that's the data itself. So you can do this fabulous all-by-all stuff, and it's housed in your data warehouse. And the problem is it's not static, it's dynamic, right? That data set's changing because of operational needs. So what are your guys' thoughts? Is that a big problem? I mean, when we think about the LHD and physics, they have that data set. Like every researcher can get to it and do reproducible research on it. How the heck are we going to do that with medical data when we have the issue that we have? Just something for you guys to comment on, please.
So, in an ideal world, we'd all share everything with everybody, because that would be swell. There are things we can control and things that some of us can't. I can't control the rights of the data that I have, because I don't own it. But what I can control is the dissemination of the evidence I generate from the data. And so I would argue that while I do not want to diminish the value and the desire, I share the desire to share the patient level data, I would say that we should not use that as the crutch to continue to make progress towards reproducibility. And so what we're kind of doing is starting at the other end. Let's say all the evidence needs to be transparent. And what we've showed is that we're now being fully transparent all the way to the point of the common data model. And we're even within the community sharing the code that transforms the data from its source to the common data model, so that at that point someone could say, well, geez, I'm all the way at the end, I might as well just share the data set. But we got to start somewhere. But I would argue that if we can get the evidence out there, a lot of what we need will be in those marginals, so we can be somewhat assured that we're making inroads towards that goal.
What about, a couple of years ago, you guys as a community did some work on simulated populations. Is that even feasible as sort of an idea that you do this large scale work on your data set and then you're able to run some kind of procedure over that and generate a simulated data set that is as similar as it needs to be in order for someone else to reproduce the specific analysis you did? Obviously, you're never going to capture full reproducibility on that data set because you can't share it. But is that even possible, or is that kind of a way out thought?
It sounds like a really good research question. I don't have the answer.
2 weeks.
So let me just comment on some of the comments towards the tail end, especially from the FDA, that you're asking us to be humble, that we have to be careful how we communicate with patients and with the community, that we don't interpret what we're doing. And so we agree with that, and we'll take that very seriously. So we understand your comments. I don't want to be so humble that we do nothing, because what we're doing now is provably wrong in ways that are actually pretty bad. And so I just want to make sure that we can get this out and have people talking and considering it. And it may be less biased than what we're doing today, even if it is. Now, we always have to be careful telling, so it always has to be couched into there's a risk and here's what we don't know. But we can't keep doing what we're doing today. So it's time for us to break for lunch.